Bismillahirrahmanirrahim. I am Dr. Javed Iqbal Kokar, Professor of Forensic Medicine and Toxicology, and I am discussing the personal identity, and this is the fourth lecture of this series. And in this lecture, I will be discussing collection and dispatch of various other steps for the trace evidences. But particularly in this lecture, I will be discussing how to collect the trace evidence. And the contents of the talk will be that I will be discussing how the trace evidence is to be collected. So collection of trace evidence, when it is being forwarded to the analytical lab, which means that in the lab it should be the study of the specimen, certain pre-lab requirements which need to be fulfilled. So before collection, we need to know what are the pre-lab requirements and steps which are to be taken. And the steps of this all processes that we have to go through: the collection of the material, then preservation of this specimen, and then finally dispatch of the specimen to the lab. So the procedure involved in the collection of material in such condition are different in different situations, and it is not the process which is involved in the clinical labs, but this is only limited to the collection of the material, collection of the trace evidence. The study of trace evidence specimen, it is divided into the pre-lab work and a lab work. The pre-lab work, it includes the general instructions. That means the instructions which are applicable to all the collection of various specimens. And there are certain then special instructions which are needed for each specimen. For example, material, if it is biological, for blood, for saliva, for semen, the special instruction for that material will be different than the general instructions which are applicable to all the specimens. So first we discuss the pre-lab work, and in this the general instructions for all the specimens. The most important step before collection is consent for the collection. If it is, you have to take the specimen from the person, which may be assailant or the victim, the consent is the necessary step. Then identification. You have to identify the individual to whom you are going to collect the specimen. Then you should know what is the source of the specimen, from where you are going to collect it. Then how to collect it, collection of that specimen. After collection, it is labeled, then it is packed and sealed, and a safe and storage should be kept so that it should not be destroyed. Then what information is to be sent along the specimen to the lab where it is going to be sent. Then the means of transportation, how it is transported from the collection to the lab. And the most important step is the chain of custody. That means the secret and sure chain of custody should be maintained. I will discuss later on. So regarding the consent for collection, consent means to say yes, to submit for examination, to submit for collection. In the living person, this consent is usually taken by the police or the magistrate. They get it in writing and present to you that the individual is ready for the collection of the material. If it is a living person and the material is collection from that individual, not from the scene of crime. This consent should always be written, and it should be signed, or it should have the thumb impression also. But if the person refuses to give the consent, then you just have to take a note it down, and then send the person back to the authority who has referred it to you for the examination. And in that cases, the specimens are usually collected by the police, but if it is for you, you have to inform the police that such and such steps have to be taken. Now regarding the identification of the individual. Identification is most important in both in living and dead, and which is done by the subjective method, the objective method, and the third party. Then two identification marks are noted. Now, regarding the source of the specimen, from where it is to be collected. Regarding the source, the important steps which are to be cared are that you should first gather the complete history of the case. Apparently, it looks alike, but when you go into detail, complete history will help you that which specimen is to be collected and from where. Then identification of the specimen. You should identify which specimen is to be collected. That means what is to be collected. You should identify, you should know, and it is only possible when you go through the detailed history. Then the proper site of collection. That means from where it is to be collected, and you will know, you should know it. Then the knowledge of character means that how it is to be collected. The person who is going to collect it, he should know the procedure, how he has to collect it. Then the method of collection, how it is collected. Because if the due care is not given for the collection of the specimen will become unavailable. Unavailable means specimen is there, but it is not fulfilling the need, the objective for which it is going to be collected. Hence, being available, it becomes unavailable. Now regarding the collection of the specimen, to ensure the collection is proper, it is advised that the process of collection should be divided into various stages, and each phase has its own goals, its own objective, and they should be fulfilled. Generally, these phases are, the first is the overview phase. You should have an overview of the whole incidents, whole process. Then search. You will search during this phase from where, from how you are going to collect it. Then the phase of recovery. You are collecting, and this is the phase of collection, phase of recovery. Then the phase of review. When you have gone through all these phases, overview, search, and recovered, then you again overview the situation so that nothing should be left. Now we'll discuss each phase, the overview phase. In this phase, the examiner go through the history, the study of documents, and may also visit the scene of crime. So it's an overall view. First, he goes into detailed history, because apparently every case seems to be similar, but when you go into detailed history, documents which the police has presented to you, and when you go to the visit of scene of crime where the incidents have happened, then an overall view you gather that how you will proceed. And you should keenly have an overall view of the scene of crime, position of the body, location of the object. So all the things should be seen and looked carefully, that what scheme of action you will be following. Then the phase of search. In this phase you will search and concentrate on the areas of interest which are important and from where the evidence you can collect. So that will be the steps now you are going to take, that which specimen is needful, will helpful, and you should collect that which is leading that case. You can move the things, move the body upside down, you can see the underneath of the body. So a detailed search should be carried out, and whatever is relevant, whatever is important, you will get an idea and what is to be taken for the evidence. Then the phase of recovery. In this phase, this is actually you recover the evidence. You collect the data, you collect the things for the evidence. That is, the specimens are collected, and each item you should identify, you should label it, and where it is located. You will write down after its identification where it's located, and if possible you should take the photograph. And this is the specimen which is lying here at this place, and I am going to collect this as an evidence. Now each item which you have collected will be labeled and should be preserved so that it should not be destroyed, mutilated, putrefied, or damaged. And then it should be packed for the dispatch to the relevant lab. Now the phase of review. You will review all the things, because if something is left, it will be left forever, because the scene of crime will be washed, it will be changed, it will be handed over to the owners to whom it belongs. So you should have a detailed review of the situation, that everything which was necessary in this case has been collected as an evidence and nothing is left. So in this phase, go through all the above phases again. And that is phase of overall review, overview or search and collection, recovery. And ensure that nothing is left, because once it is left, it will be left forever. And you may miss some important evidence. So you should review all the phases. Now what the collector should know. The specimen collection, what should we know? The collector should have complete knowledge of the choice of specimen, that which specimen is necessary for the evidence in this case. Then site of collection, from where the best material is to be collected, and what is the procedure for its collection. Then the types of preservatives which are used, because you want to preserve till it is a laboratory for the examination. It should not be putrefied before it reaches there or damaged. So it should be preserved properly. Then the container in which it is put, the size and material should be such that it should not damage, it should not interfere with the specimen. So the specimen they may be present in three forms usually. They may be either in the dry form, or they are wet form, or sometimes they are mixed with other articles. And when it is dry form, the procedure adopted for its collection is that you will scrape the specimen and make a funnel of the glazed paper, and then you can transfer it to a test tube or any other utensil. Or you can soak the cotton in normal saline and apply over the dry stain, then dry this swab in air, and this swab is taken as specimen because it contains on the surface what you have rubbed. Then if it is wet form, then you should dry the specimen and then scrape it or wipe it with wet cotton swab as the previously discussed. And when it is mixed with other articles, if it is a cloth, you can cut that portion and separate it. Now, what containers should be used for the preservation? The material of the container should be appropriate so that it should not interact or it should not temper, it should not dissolve or add some other material, which means it should be chemically inert. Generally, the glass container is preferred, but being fragile, it can be broken during transportation. So you can use plastic containers with lids, and they are preferred. Then the size and shape of the container should be appropriate, means that the material which you are putting in it should be comfortably, easily put into it, can be put into it. So thank you very much. This is the all about the trace evidence collection. Now in the next lecture we will proceed for the preservation and dispatch.